Okay, we are going to pick up with chapter 28. When last we left, we were talking about how there are many different hypotheses of how to divide up protists. One of the current ideas is to divide all eukaryotes or eukaryotic species into four supergroups, including the protists. So we said that those supergroups are excavata. So up here, we're going to talk about some of these groups. The SAR group, which includes straminophiles, alveolates, and rhizarians. We'll talk about some of these. Archaeplastida, which include some algae and also plants. So you can see plants, which are not protists. The other thing to notice that there are some algae groups up here as well. We have Uniconta, which includes, again, protists. In this group, we have protists down here in the yellow, and then that also includes fungi and animals, which are not protists, as we know. So we're going to talk about just some of these things. There's a lot of them. And again, we will look at examples. And again, keep your ear open for future changes to all of these groups when we learn more information about them genetically and how they're related to each other. So we're going to start with the excavata, as I said. So excavata is a clade that's characterized by its cytoskeleton. These protists have modified mitochondria, which we'll talk about, and some protists that have unique flagella as well. The reason that this clade has this name is because they have an excavated feeding groove on one side of their body. So the excavates or excavates, depending on who you ask, include three monophyletic groups. The diplomonads, we're not going to talk about this, but we will give examples of the parabasalids and the euglenozoans. Euglenozoans is the other group that's a part of the excavata clade. So again, these are the three clades that are three monophyletic, so three phyla that are a part of our clade excavata. So diplomonads, parabasalids, and euglenozoans. We're only going to talk about the parabasalids and the euglenozoans. So when you're studying for the test and when you're thinking about what you should know, let's think about remembering these specific examples that I talk about in the lecture. So starting with parabasalids, they have reduced mitochondria called hydrogenosomes. These are modified mitochondria, and I'll show you what those are. These hydrogenosomes are able to generate some energy anaerobically. So remember, anaerobically meaning without oxygen, so AN without. The example that we're going to talk about is Trichomonas vaginalis, which is a sexually transmitted parasite. So let's talk about the hydrogenosomes first, and then we'll come back and watch this short little video about this parasite. So this is a picture of it down here. You can see again, these are flagella. They look like they have a membrane on the outside here as well. So parabasalids. So hydrogenosomes are, again, modified mitochondria, double membrane bound organelles, just like our mitochondria, our chloroplasts, and our nuclei that are going to produce ATP using pyruvate as their primary substrate. So think back to cellular respiration. Long ago when we talked about that, remember pyruvate is the original product of glycolysis. So if we think back to glycolysis, we start with one glucose molecule. We go through 10 different steps, these conversion steps with enzymes in glycolysis, to produce two pyruvates. Those two pyruvates are going to be shuttled into different pathways depending on the organism. So if you're talking about an organism that performs fermentation, like yeast or bacterial cells, they would shuttle their pyruvate into a fermentation process, whether that be lactic acid fermentation or alcohol fermentation. An aerobic organism would take those pyruvates and shuttle it into the citric acid cycle or the Krebs cycle, which would then ultimately go through the oxidative phosphorylation step. If something was performing anaerobic respiration, still would shuttle into the citric acid cycle or the Krebs cycle, and then into the oxidative phosphorylation step at the end. So again, pyruvate is that nice intermediate that allows that organism to continue making ATP in different ways. Hydrogenozones are different from your mitochondria because they do not have DNA. They do not have cytochromes. Cytochromes are essential for breaking down toxins, and they will not, again, be using the citric acid cycle as well. And move this out of the way so you can see and I can see. So these hydrogenozones are going to contain enzymes that are capable of producing molecular hydrogen. So instead of producing carbon dioxide, like we would see in organisms that are performing aerobic cellular respiration, carbon dioxide is the waste product. In this case, it's going to be hydrogen. That's the waste product. So interesting. So let's watch this.
Trichomonas vaginalis is responsible for a sexually transmitted infection known as trichomoniasis. And as we'll talk about in a minute, the symptoms mainly occur in women in the vagina. Now, this STI is not caused by a bacterium, but rather a protozoan, which is a more advanced form of microorganism than a bacterium. And this protozoan
Okay, I think I wasn't showing the video, so I'm going to start this video again.
Trichomonas vaginalis is responsible for a sexually transmitted infection known as trichomoniasis. And as we'll talk about in a minute, the symptoms mainly occur in women in the vagina. Now, this STI is not caused by a bacterium, but rather a protozoan, which is a more advanced form of microorganism than a bacterium. And this protozoan specifically is a parasite, and it exists mainly by eating cells that they destroy, so eating cell fragments. And this protozoan uses these cell fragments or these pieces of cells for nourishment. Now, they typically have a pear shape to them, so I'll draw this pear shape here. So this is their body, the cell body. These trichomonas, or you can also refer to them as trichomonads, have one cell to them, so this is the one cell pear body. And they move around because of these protein strands, these whips that they have, that are called flagella. Flagella. And finally, one of the other things you might see if you take a look at this guy under a microscope, as we will in a minute, is the nucleus. So here's the nucleus. Now, because trichomoniasis is a sexually transmitted infection, we have to consider the mode of transmission, or how does it commonly spread from one person to the other. And as the name sort of suggests, this occurs only through vaginal sex. And what makes this infection very interesting is that the trichomonad protozoan can't survive anywhere else. So they can only live within the urogenital tract. Now, this means they can live within women, like in the vaginal tract or in the urethra, or they can also live in men. But the tricky thing about that with men is that they rarely have symptoms. So they're asymptomatic, which means that they're carriers of this infection. So they're asymptomatic carriers that can spread this STI to women without even realizing it. So if we take a look at this woman right here, and I've drawn her urogenital tract, so we're taking a look at a sagittal section, or if we chop them in half from head to toe down the middle through the legs, you can see here is the bladder that leads out the urethra. So here's the urethra, and here is the vagina. So the vagina. At the top of the vaginal tract is the cervix over here. And this part that I won't label for our conversation because we won't be talking much about it, this is the anus that leads up to the rectum and the rest of the GI tract. So if a woman is infected with trichomonas, they're going to spread into the vagina and spread up the vaginal walls. And we can take a look at this on a larger scale here. Here are the vaginal walls and we're leading up to the cervix. Let's say that the trichomonas is climbing up the vaginal tract using its flagella right here. I'll draw these green, and it latches on there. It'll use this type of mechanism to whip itself upward, and in doing so, it causes stress to the cell membrane that over time, as I'll show up here, causes the cell to die, producing fragments that used to be the cell that are scattered into the vaginal tract and eaten up by this parasite. Now, your immune system will recognize this, and in response to the trichomonas, white blood cells will be sent here to combat them and release toxins and try and engulf them and eat them to stop them from killing cells in our body. And as a result of this, we will get inflammation. Inflammation meaning more cell death in response to the ones that have been affected by the trichomonas, as well as a fever and other systemic responses. And that causes irritation. If we have white blood cells fighting the trichomonads within the urethra, you can get urethritis, which will cause pain when you urinate, and that's referred to as dys, meaning painful, uria, meaning urination, so painful urination. The same thing happens up here with the vagina, as well as with the cervix, because you can get vaginitis, as well as cervicitis. Irritation of these two will then make sex rather painful, causing what's referred to as dys, again meaning pain, pareunia, dyspareunia, or painful sex. And as this battle continues, you'll have more dead cells, dead white blood cells, dead protozoa falling through the vaginal tract out, producing frothy green discharge. And this also has a characteristically bad smell, or it's malodorous. Over time, this inflammation can actually cause some pretty bad effects as well. In women, especially those that are pregnant, chronic irritation or long-term irritation can cause them to deliver a baby earlier than they should. So that leads to preterm labor. Also, long-term inflammation can cause irregularities in cell formation or cell division at the cervix, over time producing cervical cancer. The idea, though, is that because women tend to have symptoms, we will catch the trichomonads earlier on and treat them, as we'll talk about. In men, there's a greater concern, though, because they're asymptomatic. So one of the things that you can get with chronic inflammation is prostate cancer, which is why, as we'll see in a few minutes, why it's important to treat the symptomatic patient as well as their asymptomatic sexual partner. Now, let's move on and talk about how do we diagnose trichomoniasis? Well, there are two main tests that we can employ. The first is what's referred to as a wet mount, which is exactly kind of what it sounds like. You take a swab of the vaginal tract. So you take a swab, let me draw that swab right here. And if we swab the vaginal tract, we'll get a sample of cells, mucus, and hopefully some of the protozoa as well. From there, you put the sample on this slide. So you put it on this slide. So there's our sample. And you take a look at it under a microscope. And this is what you'll see. There'll be some squamous cells. So these simple squamous vaginal epithelial cells, as well as these two guys, which are actual microscopic views of these trichomonads. And I think you can see here there's the nucleus, the pear-shaped body, and the flagella that are present here. The second test you can do is a culture, or just try and grow out the trichomonads on a petri dish. So here's our petri dish over here, and I've smeared our sample from this swab right there, and we'll let it grow over about 5 days. Then what you'll see here are these trichomonad colonies that are growing on this petri dish. Okay, so now that we've diagnosed trichomoniasis, how do we treat it? Well, here too, we're going to use antibiotics, and we can use the culture up here to determine what antibiotics the trichomonads will be sensitive to. So say, let's put antibiotic number 1 down there, and antibiotic 2 up here. Over time, we'll see that antibiotic 2 wasn't very useful. We still had these trichomonads grow, even though we had it on our petri dish here. Whereas down here, antibiotic 1 created this space where no trichomonads could grow. So that way we can have our culture determine what antibiotics the trichomonads are sensitive to. So we can have our culture determine sensitivity. And another important thing about treating infected patients with trichomoniasis is that their sexual partner should be treated as well. Because remember, if their partner was a male, they may not have any symptoms, but are at risk for developing diseases down the line. Now, what if we wanted to prevent trichomoniasis altogether? How do we achieve prevention? Well, the trick here, like most STIs, is to block transmission. And we do that by limiting the amount of direct contact during these modes of transmission. So in this case, during vaginal sex, use of a condom, so condoms, can greatly reduce the rate of spreading trichomonads potentially from an asymptomatic carrier to a susceptible patient. And because Trichomonas vaginalis can only live within the human urogenital tract, if we treated everyone that was carrying this parasite, we could eradicate the disease from the planet.
Excellent. All right, let's go back to our slides. So here's again a microscopic view of this parasite. Okay, so moving on to euglenozoans or euglenozoa. This is a clade that includes predatory heterotrophs. So think about what that means. So heterotrophs getting their energy from something else, and they do that in a predatory way. So they're eating other organisms. Photosynthetic autotrophs, we know what those are. They're using chloroplasts and sunlight to create sugar molecules. Mixotrophs, we talked about previously. Remember, mixotrophs use a combination of heterotrophic and autotrophic means to create molecules that they can use for energy. And then we also have parasites. So again, we just talked about an example of a parasite. The main feature distinguishing this group as a clade is a spiral or crystalline rod inside their flagella. So that's that unique flagella that we mentioned previously. This clade includes kinetoplastids and euglenids. So if you look at this, again, here's an example of a drawing of a euglenozoa. Euglenozoan would be the singular, OA would be the plural. And then here's the flagella. If you look at a cross-section of the flagella in the microscope, you can see these crystalline rods within them. So that is different. Most flagella are not going to have these crystalline rods within them, but the flagella will work in a similar way in that it will move in a whip-like fashion to allow for movement. So kinetoplastids have a single mitochondrion. Remember, mitochondria, A at the end is plural, with an organized mass of DNA called a kinetoplast. They are free-living species that are consumers of prokaryotes in freshwater, marine, and moist terrestrial ecosystems. So watery environments, which will be necessary for the flagella to allow them to move. Their organized mass of DNA, again, is this kinetoplast that you can see here in this image. They are consumers of prokaryotes. So they would fall into this predatory heterotrophs group. So predatory heterotrophs, meaning they are eating prokaryotes, and they're going to be breaking those things down into pieces and using those to create ATP. So example of one of these kinetoplastids would be parasites. So these parasites are able to parasitize animals, plants, and other protists. A good example of one of these is the organism that causes sleeping sickness in humans. So trypanosoma is the name of the particular microorganism that causes sleeping sickness. So we'll watch this video here in a second. Here you can see is a cross-section, or not a cross-section, but a microscope image, an electron microscope image of red blood cells and trypanosomes. So these are the protists. These are the euglenozoa that are causing sleeping sickness, and you can see what they look like here. And we find that, again, if we were to take a blood sample of a person that has sleeping sickness. So another pathogenic trypanosome causes Chagas disease. So let's watch this little video.
Human African trypanosomiasis, also known as sleeping sickness, is a disease caused by a group of parasites called Trypanosoma brucei. The two main types causing human disease are Trypanosoma brucei gambiense and rhodesiense. There are other types of Trypanosoma brucei which cause animal disease and very occasionally infect humans. Chagas disease, also known as American trypanosomiasis, is a different disease caused by Trypanosoma cruzi. Gambiense is the most common form of African trypanosomiasis, accounting for about 97% of all cases. It's found in countries in Central and West Africa and tends to cause the chronic form of disease. Rhodesiense accounts for about 3% of cases. It's found in countries in Eastern and Southern Africa and causes a more acute infection. Although there were about 300,000 estimated cases in 1995, intense control activities in affected countries have led to a dramatic decline in cases. In 2014, the number of cases was estimated to be about 15,000. Let's have a look at how the disease is transmitted. The parasite is transmitted to humans through the bite of an infected tsetse fly. These flies are only found in sub-Saharan Africa, most commonly in rural areas. Both female and male flies can transmit the infection. These flies tend to bite during daylight hours. Flies can get infected when they bite a person or animal infected with the parasite. Domestic and wild animals can carry the parasite and are important reservoirs of infection, especially for Rhodesiense. Humans are the main reservoir for Gambiense. Very uncommonly, the infection can be transmitted from mother to baby or through blood transfusions, infected needles and syringes, or sexually. Let's have a look at how the disease presents. There are two main stages of the disease. In the first or early stage, the parasite is found outside the central nervous system, mainly in the blood, lymphatic system, or other organs such as the heart, liver, spleen, or eye. In the second or late stage, the parasite invades the central nervous system. Initially, there can be a painful sore, also called a canker, at the site of the tsetse fly bite. Symptoms of the first stage include fever, muscle and joint pain, headaches, itching, and enlarged lymph nodes. It can lead to disease in the affected organs, such as inflammation of the heart muscle, enlargement of the spleen and liver, and eye disease. In the second stage, there can be a range of neurological symptoms, such as changes in personality, psychiatric symptoms, weakness, or problems with movement. The disease can affect sleep. Patients tend to have nighttime sleep disturbances and are sleepy during the day. This can get progressively worse, with patients sleeping most of the time. This is why the disease is called sleeping sickness. If left untreated, the disease can lead to death. The severity of the disease and how quickly the disease progresses from the first stage to the second stage depends on the type of parasite. In Rhodesiense infections, it's quite fast, usually a few weeks. On the other hand, in Gambiense infections, it may take several years. People who get infected may not have symptoms for months, if not years. When they do, they're usually in the second stage. The disease is diagnosed by laboratory techniques, usually by examining the blood under the microscope and looking for the parasite. This usually works well for Rhodesiense infections, but harder to do for Gambiense infections. The classic method of diagnosing Gambiense infections is by examining fluid from a lymph node under a microscope. Second stage infections can be diagnosed by examining cerebrospinal fluid. Blood tests that look for antibodies against the parasite can sometimes be used. There is a test called a card agglutination test that is used for screening for people with infections. This test only works for people with Gambiense infections. PCR tests that look for the genetic fingerprint of the parasite can also be used. There's a limited range of effective medication available to treat the disease. The type of medication used will depend on the type of parasite and the stage of the disease. The disease is harder to treat once it's progressed to the second stage. The medication used is more toxic and complicated to administer. So, how do we prevent and control the disease? Let's have a look at individual measures and also measures that could be done at a community level. At an individual level, preventative measures focus on avoiding contact with the tsetse flies. These include wearing appropriate clothing that covers exposed parts of the body, avoiding bushes in areas that tsetse flies are known to live. Insect repellents are not that effective against these flies, but may prevent other insect bites that may cause other diseases. These measures may not always be possible to do for people living and working in these areas. There is no vaccine or preventative drugs that can be taken to stop people from getting the disease. Intense efforts to control the disease have been led by the World Health Organization, National Sleeping Sickness Control Programs, and others such as pharmaceutical companies and non-governmental organizations. They focus on two areas: reducing the number of people with disease and controlling tsetse flies. Reducing the number of people with disease needs good surveillance, diagnosis, and treatment that is effective, prompt, and accessible. Controlling tsetse flies is usually done by insecticide-impregnated traps and application of insecticides to the resting sites of flies. Controlling the disease in animal reservoirs is generally hard to do. And that's a quick look at sleeping sickness, a neglected tropical disease that can have a significant impact on the health of individuals and entire communities. For more information, have a look at the websites below.
All right, so let's go back. Here we go. Okay, so we're going to move on to look at some information about algae, which is alternation of generations. These alternation of generations are going to be important not only for multicellular algae, but also for plants. So these are some terms that we're going to want to know that will be helpful with our plant chapters that are coming up too. So most complex life cycles of algae and plants include what's described as an alternation of generations. This alternation of generations is going to alternate between multicellular haploid and diploid generations. So some species have heteromorphic generations, and some of them have isomorphic generations. When a species has heteromorphic generations, the haploid and diploid generations will be structurally different. They're going to be unique when they're haploid versus diploid. If you have a species that's isomorphic, the haploid and diploid generations will be structurally similar. So there is some variation in how algae, these multicellular algae, are able to go between these generations. So when we're looking at this alternation of generations, it's important to remember that the diploid generation is called a sporophyte, and the haploid generation is called a gametophyte. And that is easy to remember, helps us to remember, because we know in organisms like us, Homo sapiens, the haploid version of our sex cells are called gametes, right? So gametes are haploid. They have one set of genetic information, and they are either sperm or eggs, depending on the sex of the organism, okay? So in our algae, our multicellular algae, the alternation of generations, there's two forms that we will see in these algae. They will be diploid or they will be haploid. If they're diploid and they have two sets of genetic information, we call that a sporophyte. If they're haploid, we call that a gametophyte, okay? So this example here, when we look at this picture, is the brown algae Laminaria. So the sporophyte, again, that's your diploid. The sporophyte will produce haploid spores called zoospores in this particular algae. So again, this is a example of a multicellular algae, and that's a brown algae. The spores develop into multicellular haploid male or female gametophytes, and these gametophytes will go on to produce the gametes. So again, the sperm and the eggs will be produced from the gametophytes. Fertilization of the gametes, again, so when these gametes are produced, they're going to produce sperm and eggs. They will come back together, typically in an aqueous or water environment. The sperm will fertilize the egg and create a diploid zygote, which will go on to make a new sporophyte. So if we look at this picture here, again, here's fertilization as an event, meiosis as an event. So here's your sporophyte. So this, again, is multicellular algae. So this is, again, single-celled algae organisms that are clustered together in a colony. So we describe this as a multicellular organism. So this is the sporophyte version. Again, 2N because it's diploid. So this sporophyte will go on to create zoospores. These zoospores are either female or male, and these zoospores will go on to create the gametophytes. These gametophytes, if they're female, will produce eggs. The gametophytes that are male will produce the sperm. Those come together to create a diploid organism. So here's your zygote after fertilization has happened. And that will go on to create the new fully developed sporophyte. So it's similar in a way to some more complex organisms like us, in that you see alternation between haploid variations or forms and diploid forms. Okay, so remember, gametophytes are the haploid variations. There's female and male versions of these. And then you have your diploid organism, which is your sporophyte. So let's finish this.
Today we're going to be talking about alternation of generations. And alternation of generations occurs when a species alternates between a multicellular haploid form and a multicellular diploid form. This occurs with some algae, some fungi.
Not looking great. There we go.
And all plants. Let's go ahead and start with the multicellular haploid form. The multicellular haploid form is referred to as a gametophyte. And it's going to have the N number of chromosomes, meaning it's going to have one of each type of chromosome. And that is going to alternate with the sporophyte. And the sporophyte is the multicellular diploid generation, 2N. 2N means it has two copies of each chromosome. Now the gametophyte is going to produce gametes, each of which contains the N number of chromosomes. And sporophytes are going to produce spores. Again, N number of chromosomes. So the ploidy, or number of chromosomes, or number of copies of chromosomes, is going to be N for everything except the sporophyte. The sporophyte is going to be 2N. And remember, we're alternating between the multicellular gametophyte and the multicellular diploid sporophyte. Now let's go ahead and connect these up. Gametophyte produces gametes, and the gametes are ultimately going to produce a sporophyte. The sporophyte produces spores, and the spores produces a gametophyte. Now let's go ahead and start up here. How do we actually get from the gametophyte to gametes? Well, we're going from N to N. So the process we're going to use for this is mitosis. Mitosis is going to give us genetically identical cells. And then in order to get from N to 2N, we're going to use the process of fertilization. Now we're going to take the gametes of this individual and we're going to combine them with the gametes from a different individual. And we're going to combine these gametes through the process of fertilization. So N plus N equals 2N. That's how we're going to get 2N. Now the sporophyte, the way we're going to get from the sporophyte to the spores is we're going to go from 2N to N. Now we're going to reduce the number of chromosomes by a process of meiosis. And meiosis is going to give us genetically different cells. The spores are N, the gametophyte is N. The way we're going to get from N to N again is through the process of mitosis. And that is alternation of generations.
So it's helpful to see how that works. All right. All right, so moving on. So we were looking at, so let's go back here for a second. So we talked about some of the stramynophiles. So looking at how brown algae are going through the process of alternation of generation. So we're in the SAR group here. We're also going to look at alveolates. Here, so alveolates are part of the SAR clade. So alveolates have membrane enclosed sacs, which are described as alveoli, and they are found and able to be seen with a scanning electron micron image you can see here. So electron micrograph, the alveoli, again, are these air sacs that are just under the plasma membrane. We have alveoli in our bodies as well. So think, do you know where we have alveoli? So think for a second. They are found in our lungs. So alveoli are the little air sacs that are at the very bottom of the branching of the trachea into the bronchi, into the bronchioles. At the end of those trees are these little alveoli. They look like little groups of grapes or bunches of grapes. They're important for oxygen exchange in our lungs. So the alveolates that we're going to talk about are dinoflagellates and apicomplexans. So again, here's the flagella that's embedded in the plasma membrane, but you can see these alveoli here. So dinoflagellates have two flagella. Each cell is reinforced by cellulose plates. So again, back to cellulose. Where did we find cellulose before? Cellulose is a primary component in the cell wall of plants. So again, it's a structural component. In this case, is also reinforcing where the flagella are embedded into the cell membrane. So these dinoflagellates are abundant in marine and freshwater phytoplankton. So think about marine meaning seawater, freshwater. They're a diverse group of aquatic phototrophs, mixotrophs, and heterotrophs. So again, we know phototrophs are using sunlight, mixotrophs are using consumer mechanisms and producer, and then heterotrophs are consumers as well. So you might have heard of red tides. Often when a red tide is called in an area, that means that you really shouldn't be swimming in the ocean water because there are toxic amounts of these dinoflagellate blooms that are occurring in the water. These toxic red tides kill off lots of fish and other organisms that live in the sea, and they're not great for people to be swimming in either. So here is an example of what a dinoflagellate looks like. You can see the flagella embedded into the plasma membrane here. Here's an example of a picture of what it looks like when these dinoflagellates, when these protists take off and have a great amount of reproduction. Creates a red color, so it looks like this red tide. This is in Australia, but it happens all over the world. Happens off the coast of Florida a good amount as well. So again, this would be a situation where you wouldn't want to be swimming. Also apicomplexans. So most apicomplexans are parasites of animals. Some can cause very serious human diseases, and we're going to talk about one of the examples. These apicomplexans spread through their host as infection cells. The infectious cells are called sporozoites. One end of the sporozoite, the apex, contains a complex of organelles specialized for penetrating host cells and tissues. So again, when you're a parasite, you're going to have particular structures. You're going to have a body shape or a cell shape in this case that is great for getting you into the tissues of your host, right? Because that's what parasites need, is a host to make sure that they're able to live and then reproduce and spread to other hosts as well. So Life cycles of most of our AP complexins have sexual and asexual stages that require 2 or more different hosts. So it makes it a little bit complex when we're trying to figure out how to um, stop the spread of these particular protist infections. It's difficult because not only do they infect one host, but they infect multiple hosts, and we have to think about how can we stop them in these multiple hosts. So one of the most common ones, and probably is a infection that you've heard of, is the infection malaria that happens in humans. This malaria is actually caused by the apicomplexin plasmodium. So we'll show you this example here. Malaria requires not only humans as a host, but requires mosquitoes as well. So we know that part of a very big way that we try to prevent malaria in people is by preventing mosquitoes from biting the people, because that again is part of the life cycle. It's a very big problem. Malaria isn't necessarily deadly, but is deadly in some populations and some individuals, particularly kids seem to have a higher rate of death. Older individuals have a higher rate of death. So 600,000 people die each year from malaria. There is malarial vaccines that are in the works. There was one that was approved in 2015 in Europe, but it doesn't provide complete protection. Right now, actually, there are some really interesting clinical trials that are going on with malarial vaccines. So it'll be interesting to see where they are in this process, because I know they were making some really good progress with this process. So let's watch this little video and then we will look at the life cycle.
Are you still using Google? Then I have something to show you. Ecosia is a search engine just like Google.
What if you could use genetic engineering to stop humanity's most dangerous predator? The deadliest animal on the planet, responsible for the death of billions, the mighty mosquito. Along with other diseases, it plays host to malaria, one of the cruelest parasites on Earth, possibly the single biggest killer of humans in history. In 2015 alone, hundreds of millions were infected and almost 500,000 people died. A new technology could help us eradicate malaria forever. But to do so, we need to engineer a whole animal population. This is not a hypothetical problem. The modified mosquitoes already exist in a lab. Should we use the technology? And is malaria bad enough to risk it? Malaria is caused by a group of microorganisms, Plasmodia. Very weird microorganisms that consist of just a single cell. They're parasites that completely rely on mosquitoes. Malaria always starts with an insect bite. In its salivary glands, thousands of sporozoites wait until the insect penetrates your skin. Immediately after invading you, they head for the liver, where they quietly enter big cells and hide from the immune system. For up to a month, they stay here in stealth mode, consuming the cells alive and changing into their next form, small, drop-like myrizoids. They multiply, generating thousands of themselves, and then burst out of the cells. So thousands of parasites head into the bloodstream to look for their next victims, red blood cells. To stay unnoticed, they wrap themselves in the membranes of the cells they killed. Imagine that! Killing someone from the inside and then taking their skin as camouflage. Brutal. They now violently attack red blood cells, multiplying inside them until they burst, then finding more red blood cells, and this cycle repeats over and over. Pieces of dead cells spread lots of toxic waste material, which activates a powerful immune response, causing flu-like symptoms. Among the symptoms are high fever, sweats and chills, convulsions, headaches, and sometimes vomiting and diarrhea. If malaria breaches the blood-brain barrier, it can cause coma, neurological damage, or death. The parasites are ready for evacuation now. When another mosquito bites the infected human, they get a ride. The cycle can start over. In 2015, the Zika virus, which causes horrible birth defects if it infects pregnant women, spread rapidly into new areas around the globe. It too is carried by a mosquito. The mosquito is the perfect carrier for human diseases. They've been around for at least 200 million years. There are trillions of them, and a single one can lay up to 300 eggs at a time. They are practically impossible to eradicate and the perfect parasite taxi. But today we have new revolutionary technology that could enable us to finally win the war against them. CRISPR. For the first time in human history, we have the tools to make fast, large-scale changes to entire species, changing their genetic information as we please. So, instead of attacking isolated groups of insects, why not just change the types that transmit diseases? Using genetic engineering, scientists successfully created a strain of mosquitoes that are immune to the malaria parasite by adding a new antibody gene that specifically targets Plasmodium. These mosquitoes will never spread malaria. But just changing genetic information is not enough. The edits would only be inherited by half the offspring because most genes have two versions inside the genome as a failsafe. So after just 2 generations, at most only half of the offspring would carry the engineered gene. In a population of billions of mosquitoes, they would hardly make a difference. A genetic engineering method called the gene drive solves this problem. It forces the new gene to become dominant in the following generations, overpowering the old gene almost completely. Thanks to this twist, 99.5% of all the engineered mosquitoes' offspring will carry the anti-malaria edit. If we were to release enough engineered mosquitoes into the wild to mate with normal mosquitoes, the malaria-blocking genes would spread extremely quickly. As the new gene becomes a permanent feature of the mosquito population, Plasmodium would lose its home base. Scientists hoped that the change would be so fast that they could not adapt to it quickly enough. Malaria could virtually disappear. If you take into account that nearly 500,000 children are killed by it every year, about 5 have died since this video started. Some scientists argue that we should use the technology sooner rather than later. The mosquitoes themselves would probably only profit from this. They don't have anything to gain from carrying parasites. And this might only be the first step. Malaria might just be the beginning. Different mosquitoes also carry dengue fever and Zika. Ticks transmit Lyme disease. Flies transmit sleeping sickness. Fleas transmit the plague. We could save millions of lives and prevent suffering on an unbelievable scale. So, why haven't we done this yet? For one, CRISPR editing is barely 4 years old, so until very recently, we just couldn't do it as fast and easily. And there are valid concerns. Never before have humans consciously changed the genetic code of a free living organism on this scale. Once we do it, there is no going back. So it has to be done right, because there could be unwanted consequences if we set out to edit nature. In the specific case of malaria, though, the risk might be acceptable since the genetic modification doesn't make a big change in the overall genome, it only changes a very specific part. The worst case scenario here is probably that it might not work or that the parasite adapts in a negative way. There is still much debate. Technology as powerful as gene drive needs to be handled with a lot of care. But at some point, we have to ask ourselves, is it unethical to not use this technology when every day 1,000 children die? Humanity has to decide how to act on this in the next few years. The public discussion is way behind the technology in this case. What do you think? This video was made possible in part by viewer donations on Patreon. If you want to help.
All right, so that is a great question for us to think about. So again, you have quite a complex system. There are parts of the life cycle that are inside the mosquito and part inside the human. Inside the human, you have the beginning phases where the sporozoites, which are haploid, are going to enter into the liver cell. That's where they grow and create the merozoites, which will burst open from those liver cells. And again, in the video, they mentioned that they cover themselves with the membranes of the liver cells, so they're not attacked by the immune system. They enter into the red blood cells. They create the gametocytes. The gametocytes are going to be picked up through the process of the mosquito bites. You're going to have fertilization that takes place, meiosis, will happen after we have our zygote formation to create the sporozoites again. So again, very similar in these alternation of generations between haploid and diploid organisms. All right, let's see. We're gonna stop right here and I'll pick back up in another video.